Hola, and thanks for tuning in. I want to tell you about our work on coffee leaf rust and other coffee pathogens in Puerto Rico. This work is in collaboration with Giovanna Marino and Luz Cerrato. I would like to focus on three subjects. Our field studies on the distribution and phenology of coffee leaf rust in the field, an overview of what we've been doing with fungal hyperparasites, which are potential agents for biocontrol of the leaf rust, and an update on our work on coffee fruit rot. Just to remind you, Puerto Rico is the easternmost of the Greater Antilles, an island 100 miles long, a little bit further south than the island of Hawaii, and coffee is grown in the central west mountain region. The important coffee pests and pathogens here are the coffee berry borer, which you all know very well, the coffee leaf rust, the coffee leaf miner, shown here, which you don't have but can be very destructive, and coffee fruit rot. Coffee leaf rust arrived in Puerto Rico in 1989. Supposedly it arrived before that but was successfully eradicated. Its arrival in Puerto Rico, according to the genetic study on which Tracy Matsumoto and Lisa Keith are co-authors, is not from Puerto Rico. It's not our fault, although you all know the coquille, which is from here. We've been doing field sampling of coffee leaf rust in parallel with Melissa and Luis. We sample every 3 weeks, 3 or 4 plots per site in 4 different points in the island. In each site, we look at 25 plants, 3 branches per plant, 1 high, 1 low, 1 middle, and we look at the incidence or the percent of leaves with coffee leaf rust on that branch, and we look at the severity, the percent of leaf area with coffee leaf rust. Here you can see our sites, and here Giovanna, Emily working in the rain. It's a lot of work, but we enjoy being out in the field. A lot of our students here at UPR are from the city, and so going out to the island and being in the country is like discovering another country for them. Here's some of our preliminary data. This is summarizing 2 years of data in 4 different field sites. We see slightly but significantly more coffee leaf rust incidence and severity in the dry season and the wet season. To me, this is somewhat of a surprise. I would have expected that with more moisture, the spores would germinate better, but our dry season is not really all that dry. Here's data from our field sites in Ciales and Orocovis, showing that both incidence and severity are highest in December at the beginning of the dry season. Here's data from Adjuntas, another site at a higher elevation, and for some reason the peak in both incidence and severity, at least in 2023, is in September, which is quite wet. We don't have any explanation for this. We have to look carefully at weather data to see what happened that year. Another field site in Lades, where we don't really see any clear pattern, and the distribution of both incidence and severity is more or less constant throughout the year. In Yauco, we see a peak in incidence in the wet season in both 2023 and 2022, but we see an irregular pattern of severity, and so far we have no explanation for this pattern. So what we're seeing is quite variable. It may be because the weather patterns have been irregular the past couple of years. So now I'd like to talk about fungal hyperparasites of the coffee leaf rust. They're quite common in the field. Here you can see half a coffee leaf with a lot of rust lesions, and the white spots is a hyperparasitic fungus that is growing on top of and parasitizing the coffee leaf rust. Our question is whether these have potential for biological control. The main strategy for dealing with coffee leaf rust is planting resistant cultivars. In Puerto Rico, Hurricanes periodically wipe out a lot of the coffee plants. For example, Hurricane Maria in 2017. And each time new coffee is planted, there's more emphasis on planting resistant varieties. For instance, after Maria in 2017, Limani, Fronton, Obata were the main cultivars planted, all of which are resistant to coffee leaf rust. There's also fungicides and cultural control, but so far there is no effective biological control that's in wide use against coffee leaf rust. Lisa Keith has been focusing on a wide range of fungal parasites of coffee leaf rust. We focused on only one, Lacanicilium lacanii. It's a fungus in the Cordycepicaceae family, a relative of Beauveria, the zombie ant fungus, lots of other fungi that are parasites on insects and fungi. And it's been isolated from coffee leaf rust in many countries. It's been formulated for biocontrol of pests and diseases of other crops, but it hasn't been formulated for use against coffee leaf rust or other pests of coffee. There have been a number of previous studies, but none of them have shown it to be very effective against coffee leaf rust. So two questions are, how diverse is the Lacanicilium population on coffee leaf rust pustules in Puerto Rico, and if all of them are equally effective at colonizing and parasitizing coffee leaf rust in the field? We isolated Lacanicilium from coffee leaf rust across the coffee growing zone, grew it in pure culture, and Steve Rayner sequenced the ITS and several protein coding sequence genes to make a phylogeny of the fungi. What we assumed to be Lacanicilium lacanii turns out to be 6 different species in 3 genera. This is partly because there are a lot of fungi that look very similar, and partly because the systematics of this group has become more refined as a result of molecular systematics. So one possible explanation for why previous studies were unsuccessful is that they were using different fungi that they weren't able to differentiate. So to determine whether all of these fungi are equally effective at infecting coffee leaf rust, we did inoculation experiments both in the lab and in the field. Here's Giovanna in the field spraying Lacanicilium spores on a coffee leaf that has rust lesions. In this graph, the black bars show the percent of coffee leaf rust lesions with hyperparasites after inoculation, both in the lab and in the field. The black bars are the leaves on which we applied spores of the fungus, and the gray bars are natural infection in the field. And you can see that there is a fair amount of natural infection, but inoculating the leaves with the fungi leads to much higher parasitism, which is good. Here we have incidence of infection of coffee leaf rust lesions with 6 different species of hyperparasites in 3 different field sites, comparing the dry season in black and the wet season in gray. And you can see that all of the fungi are effective at infecting coffee leaf rust, but the ones that work in the dry season do not necessarily work well in the wet season, and the ones that work in one site do not necessarily work well in another site. So the results are positive. They show potential for use in biocontrol, but it looks like the biocontrol may have to be adapted to the conditions at each site, which is obviously a complication. Here's a brief overview of our progress on coffee fruit rot, which Luz Cerrato has been working on for the last 4 or 5 years. Here you can see fruits with substantial coffee fruit rot, including green fruits, which according to the literature only get coffee fruit rot in Africa at high elevations, but that is clearly not the case. For reasons that we can't explain, coffee fruit rot suddenly became a big problem here in 2021. According to this news story, 5 to 10% of the harvest was lost that year to coffee fruit rot. Here's Luce in the field sampling. This is a paper that Luce published in Phytopathology in 2020, identifying the fungi involved in coffee fruit rot and showing associations with the coffee berry borer and Bavaria bassiana. As you would expect, fruits that are damaged by the coffee berry borer are much more likely to have rot inside. Here's a paper published by Mylene Caldwell, a graduate student last year, looking at the distribution of coffee fruit rot across Puerto Rico and associations with other types of fungi. Luce has a new paper this month in Phytopathology talking about Fusarium as an agent of coffee fruit rot, and once again focusing on the role of the coffee berry borer in the disease. And in her seminar, she'll give you an update on this work. When we're thinking about control of a pest or a disease, it's good to remember that control measures that target one pest will also affect other organisms in unexpected ways. Yvette Perfecto has some very elegant work from coffee in Mexico showing that, for example, the Lacanicilium that affects coffee leaf rust also affects scale insects on coffee. And there are a lot of unexpected relations, and affecting one organism will have indirect effects on others. We have really enjoyed being a part of the SCRI project, and we've learned a lot from our colleagues in Hawaii, and it's a pleasure to work with you all. We also enjoy the chance to get out in the field and fool around with coffee and its pests and pathogens. Thanks for listening.